What? I'm not sure I can. Maybe I will just leave it here.
Is that okay? Yeah, sorry.
Yeah, I'll try not to move too much. So as I said before, I don't know if you know this group of fungi. We work with mycorrhizal fungi. These are fungi that live in symbiosis with the roots of plants. It's an ancient plant fungal symbiosis where the fungi explore the soil, taking nutrients and water in exchange for carbon from the photosynthesis and some fatty acids. They exist in a wide variety of ecosystems, from desert to tropical forests, arable lands, and they involve most plants, 90% of the plants we know, and a diverse minority of fungi. And when I say a minority, they are in fact between 40 to 50,000 different species of fungi that form this symbiosis. But this is a tiny proportion of the number of fungi that we think exist in ecosystems, because the estimates are 2.5 million. So it's a tiny proportion that colonize roots of the vast majority of plants. Depending on the group of plants and fungi that form this symbiosis, well, they form different types of mycorrhizas. The word mycorrhiza comes from the Greek mykes, that means fungus, and rhiza, that means root. So they are fungus roots. And depending on the groups of fungi and plants that they form them, and the extent of colonization of the root cells by the fungal filaments, they can form two different types of mycorrhizas. When the fungal filaments get inside the root cells, they are endomycorrhizas, and there are two main types that I'm going to talk about today: arbuscular mycorrhizas and ericoid mycorrhizas. When the plants don't have roots, like bryophytes, they have rootlets, they are called mycorrhizal-like symbiosis. When the fungal filaments cover the fine roots and they don't penetrate the root cells, then they are called ectomycorrhizas, outside fungus roots. And these different types of mycorrhizas, they coexist in different ecosystems, but they also, in different ecosystems, there can be one type that dominate. And that is the case, for instance, in grasslands, fruit trees, moss crops, and tropical forests. The dominant type are these arbuscular mycorrhizas. Can you see the, yes. And they form these, they penetrate the root cells, and they form these beautiful tree-like structures that they are called arbuscles, where they branch, and that is where the exchange between the plant and the fungi take place. These ones are specialized in the uptake of phosphorus. If we think about temperate or boreal forests, trees and woody plants form ectomycorrhizas. Pine, oak, beech, spruce, hazel, sweet chestnut, they all form ectomycorrhizas. And in these, as I explained before, the fungal filaments cover the fine roots, and they form like these little globes that they change in color and texture. This is a root under a dissecting microscope. And these are specialized in the uptake of inorganic or organic nitrogen. And finally, in heathlands, the dominant type are the ericoid mycorrhizas. These are endomycorrhizas. They penetrate the root cells, and they form coils inside. And they are called like that because they associate with plants from the family Ericaceae, like heather, blueberries, cranberries. And they are specialized in unlocking nutrients from organic matter, which means that they give a competitive advantage to these plants in heathlands, for instance, when nutrients can be very low in abundance. So these different types of mycorrhizas not only have differences in anatomy, also in evolutionary history and in function. So why do we study them? We know that they can have different applications in restoration, crop production, berry production, but we also know that because they take nutrients and transfer them to the plants, they are directly involved in the nitrogen and phosphorus cycles. They also, through mineral weathering and exudating compounds that make agglomerates of soil bigger, they are involved in soil formation and aggregation. And for orchids and most of these plants as well, they are essential for seedling survival in the first stages and also in adult stages. But one of the most important functions that they have is that they are involved in the carbon cycle. And I'm going to explain a little bit more about that. In the last 10, 15 years, there have been studies published in top scientific papers or journals where they highlight that fungi, and in particular mycorrhizal fungi, have a main role in the carbon terrestrial cycle. This is a study that was carried out in forested islands in Sweden, and they found that 50 to 70% of the carbon in soil derived from roots and roots-associated fungi. We know that between 75 to 80% of the carbon in terrestrial ecosystems is in soil, and a large amount of that carbon comes from fungi and root-associated fungi. Also, in meta-analysis of experiments, that they found that plants that form different types of mycorrhizas, in this particular case, ectomycorrhizas or arbuscular mycorrhizas, they respond differently to an increase in carbon dioxide, depending on the nitrogen availability in soil. So they found that plants that form ectomycorrhizas could keep growing when carbon dioxide increased, despite nitrogen limitation in soil, while arbuscular mycorrhizal plants stop growing. So they found that it is important, but also feasible, to include mycorrhizal type, and in this case also nitrogen availability, to understand and predict the impact of climate change in the carbon cycle. Finally, there are also meta-analyses where they found that the effect of mycorrhizal type in soil carbon was larger and independent of the effect of other factors that were thought to be important. For instance, net primary production, temperature, precipitation, and soil clay content. And this was globally and across different habitats. They found that, for instance, habitats that have ectomycorrhizas, like the temperate and boreal forest, store more carbon per unit of nitrogen than habitats that form arbuscular mycorrhizas, for instance, a grassland. So they really highlight the importance of understanding the contributions of these different mycorrhizal types to carbon sequestration. I must say that the previous dogma was that the main input of carbon in soil was litter. But now we know that because these fungi are sinks for carbon from the photosynthesis from the plants, are the main key entry point of this carbon in soil. Sorry, I'm gonna drink. There is also important to take into account that a shift in mycorrhizal communities can influence carbon in soil, because we tend to think about carbon above ground. So tree planting is one of the strategies to combat climate change through carbon sequestration. But this study shows that planting trees, in this particular case, birch and pine, in soils that were from heathland, resulted in a decrease in soil organic carbon in soil after many years. And this is hypothesized to be due to the change in mycorrhizal communities, because heathlands have ericoid mycorrhizal fungi, and when trees were planted, these harbor ectomycorrhizas, and it resulted in a loss of soil organic carbon and higher respiration. So even if these systems were storing more carbon above ground, below ground, they were losing carbon. So it is always important to think about these shifts in mycorrhizal types. Finally, this is also a meta-analysis based on experiments in the field and in the lab, where they found that a large amount of carbon from the photosynthesis from terrestrial plants is at least temporarily allocated to the underground mycelium, and this equates to approximately 36% of the current annual carbon dioxide emissions from fossil fuels. So as I said, these fungi are an entry point for carbon into the soil, and they are a huge pool. But they also show that these different mycorrhizal types contribute differently to that carbon pool in soil. And for instance, while arbuscular mycorrhizal plants have a higher biomass and vegetation cover, ectomycorrhizal plants contribute more to that carbon pool in soil. The difficulty is that there is not really a method, or there wasn't a method developed to try to measure these different contributions, because most of the models that I was talking about, they are based on these different mycorrhizal proportions, inferring them from the relative abundances of the plants above ground. but no one has really come below ground and look at those roots. And this is what we have been doing in the last years. So can we develop a method? We have done this mostly in forests. This is where most of my career I've been working. And we have been able to develop a method that allows us to generate baseline diversity data of ectomycorrhizas. This is, I don't know if you have ever seen any of these. They are really beautiful in the roots. I would say that some of them are even more beautiful in the roots than above ground when they form mushrooms. Mushrooms are just the reproductive structures of these fungi, but because they rely on the carbon from the plant, they always need to be connected to the roots. And in this particular case, these are ectomycorrhizas, so they are covered in the fine roots. They don't penetrate, they only get in between the root cells. So we have been doing this in forests, generating diversity data for these fungi, elucidating the drivers of the diversity. We know that these fungi have very important functions in ecosystems. We need to know what are the drivers of the diversity of these fungi so we can understand the impact of changes in the ecosystem. We also were able to set up thresholds of different environmental variables at which we see changes in mycorrhizal communities. We can select mycorrhizas that can be indicators of different environmental variables. And also we saw that changes in communities can lead changes in the environment, and mycorrhizal communities can lead to tipping points in forests. But I need to go back 10 years ago now, 11 years ago, when I got to Kew. There weren't really many studies. There were studies in the 90s based on the production of fruit bodies, of mushrooms, that saw that there was a decline in some groups of fungi, particularly in mycorrhizal fungi. And they found that it was mostly due to nitrogen deposition. But we didn't really know what was happening below ground. And there were a few studies, but they were at local scales. So we needed to start trying to understand, develop a method, a standardized method that we could use across larger scales. And we started in oak forests. Oak has been always my favorite tree, mainly, evergreen oak because I was raised in the Mediterranean, but also because they were the main host of truffles. And truffles are ectomycorrhizal fungi, so they need these plants. I was a little bit scared about sampling in temperate forests because I thought that the roots could go deeper. But in fact, next time you go to an oak forest, if you remove a little bit the litter, you might see some roots in between. You just have to dig a little bit. And if you pull them out, you will see that they are covered by mycelium. Sometimes the easiest way to see them is to see this white, but you can find many different colors. And you will see that they are 90% or more of the root tips of oaks are colonized by these fungi. So going back to this, we selected sites from a network of monitor sites because the idea was that we could have long-term environmental data, soil and air pollution data, tree growth from these forests that we could link to the communities that we were looking in the roots. So we did this across Europe, from Spain to Romania and the UK. And we tried to cover a large gradient of different environmental variables because we wanted to understand the main drivers of the diversity of fungi associated to the roots of oaks. So we did this in Quercus petraea and Quercus robur. I did this with Martin Vidartondo. He's a professor at Imperial College who hosted me in my first postdoc here. And we traveled across 9 different countries, 22 oak forests. These are bigger ones, but our sites are 50 per 50 meters. It's a quarter of an hectare. We collected over 2,000 soil cores, more than 6,300 mycorrhizas. So you can imagine the 2 years that I spent collecting many samples. And we were able to use 18 environmental variables to link to the communities. We work with roots because it's where these fungi are always. You might find mushrooms, you might have, or not, because they are ephemeral structures, or some of these fungi produce below ground fruit bodies like truffles, and we might not find them, or they might produce crusts, and they are very difficult to find. So we know that if we look at them in the roots, we will find them and we will have a better picture of the community. We take in each of those 50 per 50 sites, we take almost 300 mycorrhizas that we identified individually. The advantage of doing this is that we can count them. So we can know the relative abundance, which ones are dominant and which ones are rare. The difficulty of doing this is that these communities have very few dominants, 2 or 3 species that are very abundant, and the rest are rare. And finding those rare ones is very challenging. Also, the number of root tips that you might find in the root of a fungus that produce many fruit bodies might not be linked. So you might find fungi that produce lots of mushrooms, and then you cannot find it in the roots. That is the case, for instance, of porcini. Porcini is an ectomycorrhizal fungus that you have probably seen in the forest, and it's very difficult to find. I didn't find a single root tip of porcini in those 6,000 roots. So it's quite rare in the roots. So what we do is to extract the DNA of those individual root tips of those ectomycorrhizas, and we select a barcode. We amplify through PCR a barcode that is the ITS, the internal transcriber spacer region. And we cluster all of these sequences in groups by similarity, and we consider they are called operational taxonomic units. And we consider that each of those units is a species. So it's the way we have to classify these fungi that might not have names, because we need to identify them by comparing these sequences to sequences that are already in databases that have names. And for fungi, since we know a very little proportion, less than 10% of the diversity that is out there, we find that even in Europe, 40 to 50% of the taxa that we find in the roots of these dominant trees, or in oak, for instance, we cannot assign a species names. So we really rely on fruit bodies that have been sequenced and identified to identify these fungi. We then assign names to these different ectomycorrhizas, but we also record functional traits. And by functional traits, I mean the presence of fungal filaments, of cores of filaments that allow them to explore the soil farther from the roots and transport water and nutrients over long distances, that they are called rhizomorphs. And they give us an idea about how far this fungi can reach from the root and the type of nitrogen that they can uptake, if it's organic or inorganic, and also the amount of carbon that they might need from the host to build up their bodies in soil. So they give us an idea of the functional diversity of these communities. So we found that almost 400 species associate with oak across Europe. We could find from 24 to over 80 different taxa in a site, but we know that they can be between 100 and 150, only in a 50 per 50 site, and only with one tree, which means that we are looking at systems that are monodominant in terms of the ectomycorrhizal trees, but below ground are very diverse. The dominant was an oak-specific fungus, is semelkap. I don't know if any of you are familiar with fungi. Yes, so this is semelkap. If you break the gills, they produce like milk. It's a specialist fungus, is Lactarius cayetus, and this is how it looks in the roots. We also found truffles. This is a deer truffle, and this is how it looks in the roots. In terms of the environmental, well, here you can see in the previous map, maybe I didn't mention, we were covering a large gradient of nitrogen deposition. Green, dark green, between 5 and 15 kilograms of nitrogen per hectare and year, and going to pink and red colors, the nitrogen deposition increases. This is ammonium and nitrate that comes from the atmosphere into the soil through the canopy of the trees in the water, and it comes from the combustion of fossil fuels. When we were looking at the number of taxa, this is a species accumulation curves, this is the number of soil cores that we collected and the number of taxa that we found, you can see that sites with red colors tend to flatten earlier because they saw less number of taxa. Between 25 30 kilograms per hectare and year is a clear a decrease in the number of taxa. So we found that mostly nitrogen deposition and soil pH affect these communities. So sites that have high nitrogen deposition, they have less fungi, and the dominants are more dominant. And pH is the opposite trend. Usually soil acidification can be linked to nitrogen deposition, we found that sites where the pH was very low, we would find less number of taxa. And interestingly, we found that there were certain thresholds of nitrogen deposition where we could start to see moderate changes in these communities, and this is between 9.5 and 13 kilograms per hectare and year. But after 17 kilograms, sites with more than 17 kilograms of nitrogen per hectare in year, we saw a steep decrease in the number of taxa. This, of course, can have consequences on the functions of these fungi, and I will explain a little bit later. We managed to include the results of this in the executive report of the network of monitor sites where we were working. The list of fungi that we found that increase or decrease with nitrogen deposition have been used to assess recovery of fruit bodies after stopping or decreasing the emissions in the Netherlands. And we were also included in this booklet in Plant Life, where they were assessing the impact of nitrogen in different groups of organisms, and they highlighted the impact that nitrogen has in this important group of fungi. But what about in other forests? These were just oak. What about in conifer forests? So back to 2013, Sietse van der Linde, that was a postdoc also working with Martin, started this project where we were collecting roots, not only, well, we included the oak data, but they were collecting also roots from pine, Scots pine, spruce, and beech across 20 different countries from Spain, Sweden, but covering a wider gradient of environmental variables. 137 sites, these 4 different trees, 13,000 soil cores, for almost 40,000 mycorrhizas. This has been the work of many people during many years, but we were able to include many more environmental variables to try to understand where, what are the main factors affecting the diversity of these fungi. We found, and I think now probably, Jed, you are familiar with the corona charts. 20% of the communities are formed by ascomycetes, and ascomycetes are fungi that form like truffles or little cups, but most of the communities are usually basidiomycetes. Basidiomycetes can also form truffles, but the main form that we can see is like the toadstools or the big mushrooms. So here you can see 62% of the communities. This is a chanterelle that is an ectomycorrhizal fungus, and this is an amethyst deceiver, Lacaria metistina. So 62% produce this type of fruit bodies, but 21% produce crusts, 11% produce truffles, and some of them, they don't even produce fruit bodies. This is, again, highlighting the importance. Almost 40% of the diversity of the fungi that we find in roots, it would be very difficult to find it as mushrooms or fruit bodies. What about so we knew that in oak, nitrogen deposition and soil pH was important. Nitrogen was affecting negatively the diversity of these fungi. This is an ordination, and each of these points is the community composition of each forest site. In yellow is spruce, in green is pine, in red is beech, and in blue is oak. I usually ask, I don't know if you know, I don't know what time is it, but it's almost time to go home. But I usually ask if you can see here a pattern. Icons that are closer together are more similar, the communities are more similar, and the ones that are far apart are more dissimilar. And probably, I don't need to ask, but probably you can see already the yellow and green are closer to each other, and red and blue as well, and these 2 groups are more separated. So this is a clear effect of host. Conifer communities are more similar between hosts and more dissimilar to broadleaf communities. So host, and not only host identity, also crown condition and defoliation were very important to assess differences in community composition. But we also found that there are other factors that affect the diversity of these fungi, and among them, again, nitrogen deposition. Nitrogen across hosts and across forests affect these fungi. They were also pH, mean annual temperature, foliar, nitrogen to phosphorus ratio, potassium through deposition were the key variables affecting these fungi. And what we found that was quite interesting is that these fungi, I told you before that we record functional traits, and the traits that we record is the type of how far they get from the root, if they can form hyphae or cords, and also if they can uptake different forms of nitrogen. And we find that fungi are able to produce more hyphae or less hyphae, or more rhizomers or less rhizomers, depending on the environment. They try to adapt. And this was supposed to be fixed within a species. We have observed this only over a space. We haven't seen this across time or over time, but we have seen that these fungi try to adapt to the environment by producing more hyphae or producing more or less rhizomers. And this is important because we have seen that fungi showing different functional traits respond differently to nitrogen deposition. And in here, this is the result of a threshold indicator species analysis. On the left, these are photos of the roots of ectomycorrhizas of fungi that decrease along the nitrogen deposition gradient. And on the right, you can see the fungi that tend to increase. The little symbols mean the little pine is that they associate with conifers, the little tree with broadleaf species, and if they have both, they are generalists. And here I think you can also see that the ones that decrease look different than the ones that increase. Can you see that? They form more hyphae, they have more rhizomers, they have different enzymatic capabilities, and they also require less carbon from, well, more carbon from the host, which means that these fungi that decrease are the ones that are more efficient at pumping carbon into the soil, and this can have consequences. And we know that the systems, well, also we found that fungi that associate with conifers are the more sensitive to nitrogen, while fungi that associate with broadleaf or they are generalists, they tend to increase. And we know that the system moves this way. So communities that look like this, diverse, different traits, fungi that form lots of these cords of rhizomers and lots of hyphae, and they move to something like this. Less fungi, less traits, and the fungi that are dominant, they become more dominant. But what are the consequences? We found that this shift in community composition was linked to the foliar nutrition of the trees, to the foliar and nitrogen to phosphorus ratio. And we saw that when there is an increase in nitrogen, these communities change. There is a shift in community composition. And when communities change, we find that the trees show nutritionally imbalances. And this is what we think is a tipping point in forests. A tipping point is an abrupt change in ecosystem state that is driven by an external factor, in this case, nitrogen deposition, and is driven by positive feedback loops. And this is because when in the system there is low nitrogen in a forest, the fungi that are more abundant are the fungi that form more filaments in the soil, and they retain nitrogen in their mycelium. So they maintain, it's like a nitrogen trap. There is almost a deficiency in nitrogen. When we add nitrogen into the system, at the beginning it's like a fertilizer. So the trees and the fungi grow. But at some point, nitrogen affects negatively the fungi that forms these many filaments. And when these fungi decrease, the nitrogen that they were trapping in their mycelium gets released. So it means that there is an increase in nitrogen that affects negatively these fungi. And when nitrogen is very high, phosphorus become limiting. So then trees show mineral imbalances and they allocate less carbon into the roots. So these fungi that need big bodies to pump carbon into the soil, they decrease or disappear. So we have seen that this is what is happening in 30% of the forest in Europe. Nitrogen deposition is affecting these fungi, and changes in these fungi is affecting the mineral nutrition of the trees. We are now trying to understand what are the mechanisms behind this. because there can be different ways for the communities to recover. We might find that after a decrease in this nitrogen addition, all the community, all the species can recover, or we might find that there are lag responses influenced by phenotypic plasticity, and that it will be ecological hysteresis, or there might not be a way back, and the system might collapse. So in our research, we are trying to get funding to test these tipping points experimentally and understand if there is recovery after crossing a tipping point. What's next? What are we doing now? Since 2021, Guillaume, and some of you, I don't know if you know him, he was part of your group at some point. So he's a postdoc in our group, and he has been working with the same data set, trying to understand if these fungi follow ecoregion boundaries, because there were this study where they were using fruit body data from GBIF, and they found that many organisms their distributions are delimited by ecoregions, but with fungi they didn't find a signal. So we thought that we could use our data set to try to find some patterns, and he has found that despite different groups having different distributions, most of them follow ecoregions. So fungi in forests within an ecoregion, even if they are in forests that are far apart, are more similar than fungi that are in nearby forests that belong to different ecoregions. And this is independent of climate and host. Hopefully he will publish the results and you will be able to see a little bit more. Also, all of these things that we have been studying, we have done it across a space. So we infer that changes in communities happen over time, but following environmental gradients, inferring that from environmental gradients. So we assume that the fungi that are in low polluted sites change to communities that we find in high polluted sites. But we haven't looked at this over time. And this is what we are trying to do now. We are resampling a subset of the sites that we sampled many years ago and try to see if there is change over time when the environment changes. And this is what Guillaume has been doing as well in the last couple of years. Also, we are trying to understand how different types of data, if we look at data collected in the roots, or if we look at data collected in fruit bodies, if those differ to infer the environmental niche and the distribution of these fungi. And so far, we have seen that the environmental niches that we can, the distributions of these fungi based on these different two sources of data may vary. So we need to combine them both. And they particularly vary if we are talking about below ground fungi, because those are difficult to find. And adding root tip data to those data sets is important to understand the distributions. We also have many projects trying to understand the contribution of these different mycorrhizal types in carbon. With all of these studies, I also wanted to highlight that it's not only important to look at the different proportions, community composition of these fungi matters for ecosystem function. And we have been focusing lately a lot on mycorrhizas and carbon. There have been studies that have tried to link mycorrhizal plant distribution with terrestrial carbon stocks, but the data that they have is inferred again from above ground data, and the data sets that are available in terms of how much those roots are colonized by different mycorrhizal fungi are very limited. If we look in the UK and try to find maps of mycorrhizal distributions, there is very little data, and it mostly comes from the 90s, and there are not really maps. We have the opportunity to, so the impact that plants have in ecosystem functioning is mediated by mycorrhizas, but we have very little information about their distributions. And also there is, again, a method that allows us to measure those different, the different contribution of those different mycorrhizal types to carbon. We have the opportunity to work at Wakehurst. Wakehurst is the Wild Gardens of Kew. They are in Sussex. And there is this program that is called the Landscape Ecology Program at Wakehurst, which aims to research, engage with and share the benefits of UK biodiversity. And it takes advantages of Wakehurst as a place that has 500 hectares of different habitats. Also the expertise that is based at Wakehurst and at Kew, and also they are a large-scale visitor attraction because they want to join different streams of research. And there is a stream that is carbon related, is climate adaptation, understanding how habitats contribute to climate adaptation and mitigation. Also, there is another stream of research that is hydrology, how habitats can cope with drought or flooding, pollination, the importance of diversity of pollinators in ecosystem function, and finally, how people feel when they are in different habitats. We are part of the carbon stream. These are the people involved in the team. And what we are doing is what we call a vertically integrated approach. We are adapting the methods that we use in forests to a method and 4 different habitats. And the idea is to understand how and where these habitats store carbon and how mycorrhizal fungi contribute to that carbon. But we are not only looking below ground, there is an integrated vertical approach where there is a team measuring through LiDAR and other remote sensing technologies above ground carbon. There is another team that measures the exchange of gases between below ground and above ground. And finally, us that look at roots in the soil. And we measure soil carbon and nutrient and also biological data that I will show you later. The idea is to integrate all the data in a model to understand how these habitats store carbon and how changes in these mycorrhizal types and mycorrhizal diversity affect that carbon, not only below ground, but also above ground. We measure, as I said, soil chemistry, and below ground, what we do is we collect many soil samples. From the soil samples, we extract the roots, and we classify them in different mycorrhizal types. We also assess root colonization. Here you can see, to see these fungi inside the roots, we need to remove all the color, all the tannins, and we stain the fungal hyphae inside the root cells. These are arbus cones, these are endomycorrhizas. The idea is to assess the extent of interaction between the fungi and the plants. We also identify all the fungi in the roots. We are using different methods for that. We quantify the DNA of fungi in the soil. We also quantify the biomass using ergosterol, that is a compound that is in the membranes of the cells. And we also assess functional traits. The idea is to have different approaches to measure or to get metrics regarding mycorrhizal fungi that give us information about how to link this to carbon. This is only in a subset of sites at Wakehurst, but we had the opportunity to be part of this large, you cannot read, I'm sorry. Large program is the Natural Capital and Ecosystem Assessment Program. It's the terrestrial arm. This is a program that runs over 3 years, funded by the government through DEFRA, and there are many institutions involved in this, including Forest Research, JNCC, and Natural England. And the idea is that they want, across England, they want to provide insights on how and why our environment is changing and the impact of that change. And they want to do this through intensive monitoring and using innovative approaches so they can deliver the knowledge needed to address biodiversity loss and climate change and to promote approaches to these challenges. So basically, they are trying to assess ecosystem condition across different habitats in England. And we have the opportunity to do this through the soil monitoring efforts that they are doing. So it's not only forests, also any other habitat. And we want to provide in situ mycorrhizal data that can be linked to above and below ground condition, identify below ground indicators related to this fungi that can be used as indicators for ecosystem condition, and increase the resolution of carbon stock maps using mycorrhizal type as predictors. We have been doing this for a year, and we still have 1 more year, and hopefully it will go beyond 2025. Basically, this is the number of sites that they are planning to collect this year. There are two different inventories for soil. Is the National Forest Inventory Plus. These are forest sites where they are collecting soils and measuring nematodes, environmental DNA, soil chemistry, and then they are measuring parameters above ground. But they also send us soil samples to Kew that we process to examine the roots. And the other survey is the England Ecosystem Survey. This is done by Natural England, and they cover any other habitats. And what I was showing you is that they are planning to measure this to send us samples from 400 sites this year, which is a huge amount of work. Each of those soil samples, they do it at two different depths, from 0 to 15 centimeters and 15 to 30. And in each of those parts of that soil core, we measure the same mycorrhizal variables that I told you for the other project, the proportion of mycorrhizal types, fungal communities in soil, biomass, fungi roots, identifying the fungi forming the mycorrhizas, identifying the host. We are trying to use PacBio sequencing, long-read next generation sequencing that allow us to identify the plants and the fungi at the same time. So we will be able to have a database where we can link every plant that we find as roots, we find the roots from in the soil to the fungi that they are associated with. We are measuring root biomass and fungal root colonization. We have been doing this so far in all of these sites. The different colors are the different land classes, because the idea will be to try to link or compare these different land classes in the different mycorrhizal communities. And we have already processed close to 140 sites. The team has been looking at many roots. You can see here almost 13,500 in the last year, which is a huge amount of work, hundreds of soil samples. We are sending 800 roots for PacBio, and we have identified 1,400 ectomycorrhizas. So this is a huge effort that can only be done with a big team in the lab. It's good that they seem to be happy, because they look at roots every single day. And the thing that with soils, you really need to be enthusiastic, because you never know if there will be roots. You never know if the roots will be alive. You will never know until you prepare them through a long process if they are colonized or not. And you can find, the fun part is that they can find anything as well, little animals that they play around with, but it is really, it takes a lot of time to go through all of these roots. So thanks to them, this is possible. And just to give you a very brief insight into how the data looks like from a subset of the forest sites. You would think that when you have trees, or in all of these models, when it's a broadleaf forest, they assume that 100% of the roots are ectomycorrhizal. We are finding here in dark orange ectomycorrhizal roots, in light orange non-ectomycorrhizal, that the range of proportions might vary a lot. And it's not only because we might have endomycorrhizal trees, also because in soil, we will be looking at roots of the understory. And many of those roots will be endomycorrhizal. Now the idea is to try to understand how these proportions link to carbon in soil. And so far from these sites, we didn't find a link between the proportions and carbon to nitrogen ratio, for instance, in soil. But we have found a link between the biomass of the ectomycorrhizal roots and the carbon to nitrogen ratio, which aligns a little bit with the inferences from above ground mycorrhizal types proportions. And this is just to give you an idea about how a network of plant and fungi identified through the roots might look like. The big nodes, color nodes, are the hosts, and the small ones are the fungi. So we will be able to know, to assign host specificity to every fungus that we find, but also to see how these communities are structured across England. I don't know how I'm doing on time. Am I okay? It's a couple of slides. The ultimate goal of this program is to have an influence in policy. In our particular case, we want to provide information about how to improve ecosystem assessment in terms of or regarding carbon stocks or nutrient cycling, but also trying to find ecosystem metrics regarding these fungi that can be used as below ground indicators. They are particularly interested in finding indicators of soil health. They also want to improve decision-making for land management, conservation, and habitat prioritization. And finally, the most important thing is that we are setting up a baseline that can be used for long-term monitoring. The only way to assess the impact of change is to have a baseline. So at least during this year, we will be providing a baseline for mycorrhizal fungi for these sets of sites. And this is just briefly to show you that we were also involved in an experiment in Wales. This is the Carbon Community. It's a charity that is interested in knowing how to enhance carbon sequestration through plantations. So this is a plantation where they established different blocks of treatments, and they planted a mixture of broadleaf trees and Sitka spruce that is quite regularly planted in Wales. And they applied inoculum from mature forests of the same tree composition and also basalt. And they are trying to see how these different treatments and the combination of those treatments affect tree growth and carbon sequestration. And we are collecting data below ground in the roots to try to understand if, for instance, inoculation of the trees with soil from mature woodlands works. And this is my almost last slide. I see that I have another one. So the idea, the next steps is to test for forest tipping points experimentally. We are planning to do experiments where we add different nitrogen and phosphorus to try to see if we can move communities to cross tipping points and we can recover them. We have this project with JNCC where we are trying to explore the value of mycorrhizal fungi as indicators of recovery after air pollution in heathlands. And we are also working in woodland carbon code sites and in plantations and natural regeneration woodlands to see differences in their mycorrhizal communities and how they contribute to carbon sequestration. So I have many questions. Some of them are, can communities recover after crossing the tipping point? We still don't know. How long does it take to cross these tipping points? Because the tipping points that we know from literature are above ground and you can see how change happens, but soil is a big black box. We never know how long it takes or when it happens. Can we measure, predict, and manage the contribution of these different mycorrhizal types? Can we really do that? And can we do it across habitats and at different scales? And ideally, can we optimize below ground management for decarbonization? So these are my questions. I wonder if you have some for me. Thank you. That is the team at Kew.
Great, thank you very much for a fantastic overview. Very educational in lots of ways. I'll kick off with a question and we can hand over to others. What are the roles that mycorrhizae are proposing is to, as inoculation, to facilitate restoration? And I know there's some controversy around this. I was wondering what your thoughts are, how useful fungal inoculation can be a way of facilitating, enhancing restoration of ecosystems.
So, because we tend to think that all plants can associate with all fungi, and that's not the case. For instance, if the plan is to plant trees in a grassland, the fungi that are in the roots of the grasses are not the fungi that associate with the trees. So in that particular case, it would be probably useful to inoculate the seedlings to accelerate the process for them to establish. But if there are forests around, or if there are edge rows that have similar composition than they have ectomycorrhizal fungi, then the inoculum will probably be enough for the trees to establish. There are very few, so there are experiments where they have done that in pots, and they can see the beneficial effects of inoculating fungi, but there are not really many field trials where they have tried that. In areas where the soils are difficult or there is no natural inoculum, it can accelerate at least the process. In this particular plantation, there was an effect of inoculation in tree growth the very first year, but that effect seems to be decreasing. But we still need to know a little bit more.
Thank you. We can open up to questions. At the back there, Andy. Thank you very much for a great talk. That's really interesting results on the negative effects of nitrogen. Obviously said a little bit more about that, but I just wonder if you could say more about the mechanisms of results. Do you think what's happening is that the tree saying, I'm trying to see you get nitrogen now, I don't need you anymore, it's kicking the mycorrhiza out, or something else? Or?
I think there is a direct effect of nitrogen in the fungi, that because nitrogen can be toxic. But there is also an effect of less carbon allocation to the roots when the nutrients are directly available for the trees. So there is both ways, because this fungi, what happens is that because they rely on carbon from the plant, and some of these fungi need a lot of carbon to build up those bodies. If nutrients are already available for the plant, the carbon cost for the plant is not efficient. So we even think that some of these fungi might be almost be parasitic. They might not even be doing much, or they might have other functions like gathering phosphorus. Because when there is a lot of nitrogen and CO2 is increasing, trees tend to grow, but at some point they need also more phosphorus, and phosphorus become limiting. So associating with fungi that are able to explore the soil for phosphorus might be an option.
Waiting for the microphone. Thanks very much. I really enjoyed it. Is it working? Okay.
Yes.
I was amazed that you had so much diversity of species of mycorrhizas just on a single plant species. So you've got a single oak tree hosting enormous numbers of different species. That's really extraordinary. Any idea, how that coexistence of all those different fungi occurs? And do you see in a more diverse woodland with many more tree species, do you get a much more mycorrhizal diversity in total, or is that not really that important given the extraordinary diversity that each individual tree can support?
Okay, regarding the first question about the diversity, I guess that there are many different niches that these different fungi can occupy. I don't know if it can explain such a great, because some of the traits that they have, they are shared traits, so there is functional redundancy. But these different fungi have evolved many times to being ectomycorrhizal, and there was a big diversification when there was a diversification of the angiosperms as well. In terms of if more diversity might end up in more fungi, it will depend on the composition. So some of these fungi are specific to host, but other ones are generalist. So you would tend to think that when there are more different hosts and far apart, you might find more fungi. I did a study looking only at pine roots when it was co-occurring with oak or not, and I found more fungi when it was co-occurring with oak.
Even on the pine?
Even on the pine.
Thank you.
Obviously, I guess one of the big sort of reasons why mycorrhizal fungi have gained more attention recently is because of the fact that sometimes they seem to allow trees to connect with one another. I guess what I wanted to ask is how does the possibility that mycorrhizal fungi form networks between the trees affect the things that you're studying, especially I guess community composition and things like that. With the studies that you are doing, is it possible at all to assess sort of how these mycorrhizae are connecting trees together? And if so, what effect does that have for everything that you've been studying?
So there's a lot of controversy
Yeah.
about these mycorrhizal networks right now. It's more about how permanent they might be in a forest, for instance, because so in a root, when one root tip is colonized by one fungus, there is a high chance that the next root tip will be colonized by the same one. When we look at the different root tips colonized by these fungi, we assume that is the same. It can be the same individual or it can be a different one, because we are only looking at a small fragment of that DNA that might not give us that information in terms of if it's the same individual or not. Usually the ones that form these networks are fungi that are generalists, so they can associate to different hosts, or specialists if the two plants or three plants that are connecting are of the same tree species. In our particular case, we don't focus on individual trees. We do transects, so we cannot really link which tree go with which fungi. We have done that with the small plants above the tree line. You know that there are some dwarf willows that are also ectomycorrhizal, and you can find them. And for that, it might be, well, depending, because some of them, they also like are creeping willows, so they extend below ground and you cannot really see, but it will be easier if the plants are smaller. But in the case of trees, it's very different unless you genotype the roots as well. So assessing if the same fungus is in two different trees, we only know that the same species is in two different trees, but we don't know if it's the same individual, because some of these fungi as well, they only spread through the roots. Well, they spread mostly through the roots. But we will need to do more population genetic studies to know if they are the same individual or not. I don't know if I responded to your question.
So I had thank you very much. I know we've talked about this before, but I had a similar question around the controversies around the common mycorrhizal networks, or the wood wide web, as sort of a lot popular romanticization has kind of brought attention to mycorrhizas. And I was wondering if you could maybe comment a little bit on the benefits that this popularization has brought to the mycorrhizal ecology community, and some of the drawbacks that some of this attention has also kind of brought to.
So benefits, people are more aware of these fungi. When I started, it was very difficult to get projects related to mycorrhizal fungi. People are interested about other things, and looking at roots is boring. Also, there is some work behind those studies that was I'm gonna say well done. It sounds like a burger, but well done, or a steak. But the problem is that many papers, instead of saying that there is a potential for those networks, because it is clear that the transfer of nutrients happen, because if a fungus is connected to two different plants, the fungus is transferring nutrients. It is not that clear that carbon can move that way, only in particular cases where, for instance, there is shade and the seedlings cannot produce carbon to transfer to the fungi, so there might be some allocation from the next tree. The disadvantage is that people tend to reference only studies where that was, not only studies, that tend to assume that that is the case always, when that can only happen in certain circumstances. And also because they assume that those networks are more permanent than they might be, because the probability probably of having such a big network under the soil when there is a lot of disturbance, and it might not be possible. They also think that there are alternative explanations to the results observed in those studies, so people need to be careful and say there are potential networks instead of assuming that they are already existing, basically. I don't know if I have
Oh, yeah, that's great. Thank you.
But basically, I think people know much more about mycorrhizal fungi, and they are much more interested as well based on that pioneer work. I guess that is not generalizing more than anything else.
Not going to trust that this thing works.
Online.
Hysteresis and tipping points. My question is about that. Well, you had a slide that had systems that were kind of healthy and stable. Then you had the hysteresis thing, and then you had the collapsed. Now, I think of hysteresis as a system that's kind of stuck a bit because of its past it's very memory dependent. But it's kind of hopeful because if you want to move it from one state to another, if you put some force in, you might be able to move the state. But I'm thinking tipping points is where you've gone to something, you've gone beyond it, and you're now in a phase where it's self-fulfilling, and it's really difficult to actually fight against that. And that's kind of a bit less hopeful. Now, if you're setting up a natural capital committee, I'm guessing what you're doing is you're wanting to look around for systems in order to invest, to look for those systems that are before a tipping point but are close, and you want to keep them away from the tipping point. And you don't necessarily want to be investing where it's way beyond it and there's no hope. So can you give us some insight on what you mean by these terms? And if you do want to sort of have a model to invest in nature, how you would, whether this is ever going to be good enough to do that, basically.
Well, this is based on space, not time. So this is a hypothetical tipping point that we think is happening because we generated a large amount of data. So the idea is that you're right, hysteresis is when the system goes through a different state on the way back, right? And sometimes you have to push the conditions that led to a tipping point farther back on the gradient. So it doesn't mean that the system is gonna recover exactly in the same point where it changed. That's my understanding. We think that these fungi, because they are able to adapt, phenotypic plasticity has a role in that recovery, and might push the threshold at which change or recovery happens earlier, but it can also push the threshold at which changes happen farther. In terms of where to invest, we also think that if we could predict when that tipping point as well is not only just one point, it's like a domain. And we predict that that tipping point happens at a certain place in the gradient, but we haven't tested it experimentally. Where we think is that when you are close, when you have just crossed the tipping point, there might be more hope for recovery. But if you are beyond the tipping point, recovery might not be possible. So the experiments that we were trying to do was to push systems, so find sites, and I'm not sure if I'm responding again, but what we were trying to do is that find sites that are before the tipping point, in that domain, and then after the tipping point. Add nitrogen to push them towards crossing the tipping point, and add phosphorus to alleviate phosphorus limitation to try to push them back and see where those changes happen. But I guess that you have more probability to recover a system when you are closer to the tipping point.
Thank you. There's a couple of questions online that I'll convey to you as well. There's one that I'll combine two people. So Britt Pugh asks, the insights that you've had on soil carbon storage in European tree species, how might these apply to tropical tree fungi associations and soil carbon? And Walter Horaka, one of our former lab members, asks a little bit about how the same methods can be applied to arbuscular mycorrhizae, and is your project interested in expanding to tropical ecosystems as well?
We are expanding, right? So soils in tropical ecosystems I don't have experience with soils in tropical ecosystems looking at carbon. They are differently structured, and the organic layers are, I think they are thinner. And the nutrient that become limiting is phosphorus. So I think the dynamics are slightly different, but in these studies that I was showing, they also sample tropical forests. Tropical forests are usually, there are some exceptions with Dipterocarpaceae, but are arbuscular mycorrhizal, which means that the dynamics are slightly different than the ectomycorrhizal. And in fact, under nitrogen deposition, arbuscular mycorrhizal plants might have an advantage against ectomycorrhizal plants. And we have seen, or researchers have seen that in North America. But I don't have a lot of experience on tropical systems, but we are moving forward to do that. We might have a grant coming, looking at soils, and I also have a project in Madagascar, and we have worked a little bit in the Valdivian forest in Argentina.
Right. Okay. Yes. Okay. There's
But
one.
there is still lots to do in temperate ones.
Yes, yes. And tropical systems are often quite abundant in nitrogen supply. So you can see the advantages that you talked about there, the vascular systems seem to have an advantage there. And there's a second question by Will Brown. It's a bit long, I'll try and paraphrase it. Do you know what is happening to the below ground carbon cycling of a mature tree that is starting to slow growth? It's still a similar photosynthesizing surface area to produce sugars, yet less resource requirements as it slows growth. Current carbon models that look above ground predict carbon sequestration slows, but could there be a role for below ground carbon cycling? Yeah, because so the trees are still photosynthesizing a lot, but they're not growing so much above ground. Is it because something is shifting below ground?
There are cases when I'm not an expert on this, but there are cases, I talk about the nitrogen trap. So there are cases where these fungi, when nutrients are low and these fungi retain nitrogen in their mycelium, they also maintain the system almost in a stress. So they might restrict growth of the trees.
Right.
It's called the nitrogen trap. And well, this gets released when there is a nitrogen addition in the system. That could be an explanation. I also know that some ascomycetes, like truffles are little beasts. They are very competitive and they live in very tough environments. And they also tend to maintain sometimes the plant in a certain level of stress where they still get carbon, but they don't really make these trees grow. So there might be, in some cases, where these become less beneficial for the tree that in a mutualism should happen.
Okay, great. Okay. No more questions anymore? Oh yeah.
So I guess I was thinking about below ground biodiversity. You've obviously got the plant roots and the ectomycorrhizal fungi. And I was just wondering if you or anyone else is, I don't know if there are any other big players that are part of it, you know, the bacteria.
Saprotrophs, other fungi.
Yeah.
Yes,
What other dynamics are going on?
They are not the only ones, but they are very important and they have been overlooked for a very long time. In fact, these ones, all the fungi have important roles. Decomposers are very important in ecosystems. Otherwise, we would be covered up to our ears in litter and leaves and all of that. But there should be some sort of balance between abundances of these different fungi. Also, decomposers tend to release carbon dioxide into the atmosphere, and some of these ectomycorrhizal fungi are able to compete with them and slow down decomposition. But bacteria and many other organisms and other fungi are important. It's not just these fungi, but these are the ones that are the link between the carbon from the plant to the soil and the ones through which the nutrients pass to the plant. So in our group, we are focusing on this one.
Can I ask what you do with all the truffles you find?
You know, when I was doing,
You don't.
when I was doing my PhD, the problem is that, I have been always working with roots. So I didn't get to collect truffles, but I was attending many conferences where we would eat them, many. In fact, maybe I shouldn't say this because this will be in YouTube. I sent a couple of truffles to my mom. She didn't really know. Many people tend to think that truffles are like the chocolate truffles. So I sent them two very expensive ones because the kilo is very expensive. And she called me back and she told me, Laura, these are worse than potatoes. And I said, but Mom, why do you say that? Well, I just cooked them with beans and chorizo and they didn't taste that much. So because you know you need to put them with things that don't. So I didn't get to eat many. But at that time, what we were trying to assess was why there were plantations that produced truffles and plantations that didn't, and why different trees also have different production. And at that time, we thought that it was related to the amount of mycelium in the soil. Now we know that it's not just biomass of mycelium, it's mating types that need to need to reproduce, basically.
Great. I think we can end on that question. And thank you once again, Laura, for fascinating education there and the insight into the world of mycorrhizal.
Mycorrhizal fungi.
And feel free to join us for a drink around the corner as well. So, thank you.
Thank you.